So let us begin the presentations on livestock and climate change and overview. And uh, this is my presentation flow. I'll be talking about a little bit on the climate change, what it is, and what are those factors which are leading to the climate change, and the impact of climate change, particularly the heat stress, because most of the time it is the temperature, apart from the other factors. The temperature is much more important than the other factors. Then greenhouse gas emissions and human methanogenesis, whether it is a necessity in terms of the metabolic functions, or this process is purely a waste process. So we'll look into that, and what is the methane emission scenario, the globally as well as in Indian conditions, and what are the possible mitigation approaches, majorly focusing on the mitigation approaches what we have developed here at NNP, and which are much more farmers friendly from the Indian perspective. And then what would be the implications in terms of the productivity enhancement, or in terms of the minimizing the impact of climate change on the livestock productions and the farmers, and in the end conclusion. So let us see what the climate change refers to. Basically, it is a long-term shift in the temperature and weather patterns. Day-to-day fluctuations, we cannot call it the climate change. The day-to-day variations are something seasonal and temporary. But when you say the climate change, it is a permanent phenomenon which happens in the long term. And this long-term change in the climate could be due to the natural factors, or mostly it is anthropogenic. Anthropogenic means man-made factors are mostly responsible for the change in the climate. So this climate change impacts, if you see, it is leading to the melting of glaciers, the drought, flooding, hurricanes, and the fire, and so on and so forth. And the implications or the impact, if you see, it is mostly the climate change is happening because of the greenhouse gases, the large emissions of that, and it is leading to the loss of the most of the biodiversity. Here you can see, so almost 20 to 30% biodiversity is already lost because of the climate change. Then if we look at the factors, there are two factors which are categorized into natural and human induced factors, which you call the anthropogenic one. If you see the natural one, it is the changes in the solar output particularly, and so and the solar output means it is the referred to the solar cycle, basically solar cycle, which is a natural phenomenon, and it happens almost every 11 years, and this refers to the changes in the magnetic field of the sun. So it affects the solar radiation output from the sun, basically. So it is a natural phenomena. And then change in the Earth's orbit, and we know that Earth is tilting towards the sun and its own axis. So the tilting as well as the incline toward the axis refer to the, or leading to the change in the climate, the warming as well as the cooling. And the aerosols. Aerosols are the minute particles which are suspended in the atmosphere, and basically what they do, they absorb the radiant heat and leading to the warming of the atmospheric surroundings of the Earth. If you look at the human factors, it is mainly the greenhouse gases, and which are being produced due to the anthropogenic activity. So anthropogenic greenhouse gases are the major and the foremost factors leading to the climate change. Apart from that, the land use change, particularly the deforestation, is the one which is majorly responsible for the change in the climate, and the atmospheric aerosols and the burning of fossil fuel. Fossil fuels is majorly related with the emissions of the greenhouse gases, the majorly the carbon dioxides and the nitrous oxide or the nitric oxides, etc. So then, if we see in what way the climate change will impact the agriculture system, which is mostly referred to the higher temperature. This higher temperature will lead to the variations or the changes in certain factors, like the weather conditions, the rainfall patterns, or the emergence of the new diseases, and the crop productions, biomass yield, and so on and so forth. So about 20 to 30% biodiversity, as I told, is already been lost because of the climate change. And even a small change in the temperature in the tune of 1 to 2 degree will also lead uh, to the endangered or extinctions of the biodiversity by 20 to 30%. And the livestock productions, whether it is dairy animals or the growing or the wool, particularly the sheep, they will also not remain unaffected with the climate change. So climate change, apart from the agriculture, will also going to impact the livestock production as well, and particularly through the heat stress. And we'll see what would be the mechanism, how it is going to be affected. And of course, the food security is the one. As you know, that the change in the climate or the climate change will lead to the less biomass yield, as well as there will be some compositional changes as well. So that means the overall productivity as well as the absolute productions is going to be affected. And if you correlate it with the accelerating human population, so there will be less availability of the food items, and which can lead to the conflict over the scarce resources among the human beings. And the developing countries are those which are going to be mostly impacted by the climate change. Sorry. So if you look at the impact of climate change on feed composition and biomass yield, there will be much more impact of the climate change on the C3 plants as compared to the C4 plants. And why it is so? You know that C3 plants are different from the C4 plants in terms that the first stable product of the C3 cycle is the 3 carbon compound, and in C4 it is oxaloacetate. If you see in the C3 plants, physiologists may be aware about it, that the Rubisco enzyme particularly are responsible for carrying out the C3 cycle. And this Rubisco enzyme is mostly impacted by the temperature. More the temperature would lead to the less affinity of this Rubisco enzyme towards the carbon dioxide. And the carbon dioxide is the main substrate for the photosynthesis. So the photosynthetic capabilities of the C3 plants is going to be adversely affected by the climate change. And this is how the climate change will majorly impact the C3 plants as compared to the C4 plants. And the increasing temperature definitely will reduce the water soluble carbohydrate content in the feeds, fodder, or the biomass, as well as the protein content. And if you look at the impact in terms of the high latitude and the low latitude, at a high latitude, you know, this is the area towards pole, and where the growing season is very short or growing duration. So towards high latitude, high temperature will prolong the growing season, and this is how it is going to impact the cropping pattern as well as the biomass yield. On contrast or opposite to it, at low altitude, what is going to be affected by increasing temperature? It may lead to the more evaporations, and therefore will create the water stress or water scarcity, and therefore the biomass yield. Simultaneously, the harvest indexes will also change. What the harvest index refer to? It is the grain to the shoot or stem diameter ratio. So this will also change. If you see, in case of the rice or the wheat, it is almost 0.3 to 0.5 the harvest index ratio is. So it is going to be affected by increasing diameter content or the fibrous portion in proportion to the grain. So once it is changed, the metabolizable energy content of the that particular feed stuffs or ingredient will also be changed, and there will be more incidences of the drought as well as the diameter changes in the diameter yield due to the more lignifications. So if you see the linking of the climate change as well as livestock production, it is very interesting and very complicated too, because livestock are the sufferer of the climate change as well as a major contributor too. So it is two-way basically uh, mechanism or interactions of the livestock with climate change. They contribute to climate change and also affected by the climate change. If you see how they contribute to climate change, mostly through the enteric fermentations, which release the methane as a major greenhouse gas, and they dispense this methane into the surrounding atmosphere. And apart from this enteric methane, a small portion of the greenhouse gases of methane and nitrous oxide is also being emitted from the excrement management, like the burning of the dung cakes, which was a usual practices in the rural area, although a trend has little bit changed over the years. The biogas and the dung heap. Through this excrement management, the methane and nitrous oxide is also being emitted in the atmosphere, which lead to the temperature rise or the global warming, and increasing temperature affect the water resources. So less water is available for the food and fodder production. So biomass yield is affected, as well as the it also lead to the less drinking water supply to the livestock. So productivity as well as the reproductive efficiencies, everything is being affected by the climate change. So this is how the climate change and the livestock are interrelated. Sorry. So this is the impact of the increasing temperature, particularly the heat stress, how it impacts the livestock species. So if you see, increasing temperature will lead to the high skin temperature, because the animal body surface or skin absorb the heat, and so the temperature increase. Apart from increasing skin temperature, the rectal temperature, respiration rate, and the heat rate, everything goes up. So during this course, what happens? The hypothalamus or the pituitary, they release the corticoid releasing hormones or adenocorticoid releasing hormones, which in turn lead to the more production of cortisol. So during stress, the production of cortisol goes up, which is related to the metabolic activities. So this is how, or you can say this is the physiological adaptation or mechanism which is being take place in the livestock due to the increasing temperature or the heat stress. So if you look at the correlation, it is not only the heat stress. Let me tell you, it is the humidity also. Uh, uh, if you discuss the temperature alone, it will have less adverse impact as compared to a combination of the temperature and humidity, because humidity has a cascading effect on the heat stress. So a combination of the temperature and humidity is much more adverse or consequential in terms of the impact on livestock production. The temperature humidity index is therefore calculated by temperature and humidity both. So the THI score less than 72 or around 70, we can say the livestock is in comfortable zone. They do not have any stressful conditions. But anything exceeding 72 is showing some stress. Up to 78, it is the mild stress, and from 79 to 88, it is moderate, and anything 90 and above is the severe stress. Here you can see that the correlation between the temperature and the humidity. When the humidity is high and the temperature is also high, it is showing or reflecting the more heat stress conditions as compared to the high humidity alone or the temperature alone. So if you look at the greenhouse gases, these are the greenhouse gases. Everybody is aware about these. And if you look at the concentration, it is mostly the CO2, which is being prevailing in the surrounding atmosphere. But being the animal scientist or animal nutritionist, we are majorly focusing on methane. And these are some other perfluorocarbon, sulfur hexafluoride, and so on and so forth. These are some other greenhouse gases. So if you look at the overall concentration of these greenhouse gases in the atmosphere, irrespective of the type of the GHG, it is around 470 ppm. And out of this 470, 441 alone is the CO2. If you look at the methane concentration, it is 1880 ppb. So you can say the concentration wise methane is far less as compared to the CO2. But still, we are majorly focusing on methane as compared to the CO2 or the other greenhouse gases, because the CO2 is majorly coming from the industries. However, the methane is primarily comes from the agricultural sector. And these are the concentration of some other greenhouse gases. If you look at the global budget of the methane emissions, irrespective of the type of sources, like the green one reflects the natural, and this red one is showing the anthropogenic sources. It is around 558 teragram or million metric ton of the methane is annually being emitted, irrespective of different sources. And out of this 551, 548 teragram is being removed through various things. So that means there is a net accumulation of around 10 teragram of the methane annually. So this 10 teragram keeps adding on every year to the global methane pool. And you know, the concentration is almost 1880 ppb, which is also increasing at a rate of 10 ppb every year. So that means if you have 1890 ppb in 2022, on an average, it should be 1900 ppb in 2023. So here at the NINP, we have done some exercise and tried to calculate the greenhouse gases in terms of the CO2 equivalent or the carbon footprinting of livestock production by considering the partial LCA approach. It was not the complete LCA, it was the partial LCA, where we have considered only two factors, the enteric factor as well as the manure management, and the methane and nitrous oxide emissions. So our estimate revealed that altogether it is around 260 million metric tons in terms of the CO2 equivalent is being contributed by the livestock. So now, apart from the contribution of methane in the global warming or the climate change, what are the other implications? Why do we need to discuss about methane? Is it only the global warming? No, it is not only global warming, because if you consider only global warming or climate change, the contribution of CO2 is much more as compared to methane. So that is not only the region that we are discussing the methane emissions. The much more meaningful reason or The factor is its wastefulness. What is the wastefulness here involved is, if you see, the CH4 or the methane has a carbon moiety. So when the animal is releasing it into the atmosphere, almost 95% is coming out through belching or through breathing, and remaining 5% from the large intestine, which is coming through the anal way. So this loss of methane or the dispensation of methane into the surrounding atmosphere is also a loss of the energy because of carbon moiety. So we have done one exercise and um, prepared an estimate that around 38 into 10 to the power 8 gigacalorie of energy is being lost in the form of methane, and that is true from the dairy animals, dairy cattle and dairy buffaloes. We have not considered the other species, or you can say the non-dairy animals here. Otherwise, the figures would be really very high. So we have considered the impact in terms of the energy loss in form of methane from milking animals, and it is very high. A state-wise emissions is also in terms of the energy loss from the dairy animals is also being given here. So if you look at the enteric methane emissions estimate in Indian scenario, there are various agencies which have furnished various estimates, and it is highly, you can say, the uncertain. That it is as low as 7 TG teragram or as high as 18 teragram. And you can see the variations, it is highly flip-flop. So this really creates some confusion among the researchers. And what are those factors which are leading to this uncertainty? Here is the answer for that. Most of these these estimates have used the IPCC based tier systems. IPCC tier system 1, tier 2, and now tier 3 is also available. So most of these estimates have used the tier 1, and only 1 or 2 are based on the tier 2 system. So if you see the tier 1, what this is the way through which we calculate or estimate the enteric methane emissions from Indian livestock or anywhere if you are using the tier 1. This is the emission factor multiplied by the population of that particular livestock category, and then 10 to the power 6. This 10 to the power 6 because we are calculating the emission in terms of gigagram, but emission factor is in kilogram. So if you convert the kilogram into gigagram, you have to use this constant 10 to the power minus 6. So basically, if you see what we are doing here, we have considered an emission factor single, which is 58 kg for Indian dairy cattle, and then simply multiplying with the population. So this is a very inaccurate, or you can say crude way of calculating the enteric methane emission for a country like India. There is no consideration of the feed intake, feed quality, seasonal and regional variation, nothing. A cattle in Assam may not be producing the exact quantity of enteric methane which is being produced in Punjab or Haryana or Himachal, because methane emission is a function of the intake, feed composition, and production. It is a cumulative function. So here there is no consideration. That is why the uncertainty you have seen in those. Then later on, the people also tried with the tier 2, which is based on the consideration of the gross energy content of the feed, and YM refers to the efficiency of the energy intake which is being converted into methane. But there is only one energy value considered here, and again there was no consideration for the regional and seasonal variations or fluctuations. In India, if you see, the feeding practices in Himachal is different. Jammu and Kashmir, it is very much different than what is being followed in UP or Haryana or Punjab. In South India, it is different. And even within a state also, 365 days, it doesn't remain same. It keeps changing in different seasons. So there is no considerations. And the tier 3 system is purely the modeling base. And here we have to develop our own emission factors by considering everything. So I can say the tier 3 system would be much more accurate as compared to the others. And that is why the usage of the tier 1 system or the tier 2 systems led to the uncertainty in enteric methane emissions estimates in India. So at NNP, we tried to develop an inventory, state-wise inventory, this you can see. And our estimates revealed that the livestock in India annually emits around 9.25 teragram of enteric methane. And this is the state-wise enteric methane release. And here you can say the national average is around 2.54. There are few states which are emitting more. And the emission here more is because of the livestock population as such, or more number of the bovine species as compared to the ovine. We also tried to furnish an estimate based on the emissions from different species, and our estimates revealed that around 56% of the enteric methane in India is coming from cattle, 29% from buffalo, and 15% from the sheep and goat. Of course, there are few more species, but their numbers are so small. That is why we have not taken into consideration like camel, yak, mithun. These are pig, some species which also lead to some methane emissions. So if you see, we also tried, this is the one way of comparing. The other way is, which generally the European or the Western world says or emphasizes too, that please don't compare the methane emissions as such in absolute word. Rather, you compare in terms of the output. Means how much is the methane emissions per kg of the milk productions. And here you can see there is huge variation. In a state like you see, it is around 85 gram of methane per liter of milk. On the other hand, if you can see the Punjab, Haryana, Kerala, it is around 6 to 10 or 15. So anything less than 20 gram of methane per liter of milk is perfectly fine. But beyond that, that means the animals are not productivity wise efficient, and either they do not have the genetic makeup, rather than simply or merely making the high contribution to the enteric methane emissions. So if we see how this methane is being produced, there are certain microbes which include bacteria, protozoa, fungi, virus, or the bacteriophages or the archaeophages which reside in the rumen. And all these microbes live in a syntropic association. What is the syntropic association? You see, once the feed is being utilized or ingested by the animal, it will go to the first stomach, which is referred to the rumen. And there these microbes under anaerobic conditions will act onto the feed, and then they will produce certain metabolites. So these metabolites, which are majorly coming from the bacteria, are being used by the other group of microbes, and then they produce something else. And this kind of relations is called syntropic relation. It is different from the symbiosis relation. In symbiotic relations, the one group of microbes is giving something to the other group of microbes, and in turn they are also reverting back or giving something to the first group of microbes. So that is symbiosis. But in syntropic, the end product of the first group of microbes is being used by other group of microbes, and they also produce something which is used by the third group of microbes. So that kind of the relations is syntropic association. That means all these microbes are linked and dependent on each the others. So during the fermentation, that this gases, mainly the CO2 and hydrogen, is being produced, and the methanogens combine these and release in the form of methane. So where does these methanogens occurrence? Or you can find them anywhere. Wherever the anaerobic conditions exist, these methanogens can be pinpointed there. So the gastrointestinal tract or of the animals is one of them. You can find them in soil, particularly in rice, or in oceans, or anywhere wherever the anaerobic conditions exist or been created in short time. So if you look at the methanogenesis, whether it is a metabolic necessity or a waste process, here if you see what it is removing, it is removing the hydrogen. From where? From the rumen. You see, one peculiarity or the specific feature of the rumen is the epithelial membrane is impermeable to these gases. These gases cannot pass through the epithelial and cannot go to the body system. So that means whatever these gases are being generated or produced in the rumen will keeps accumulating, because it is not passing through the rumen epithelial and has to be necessarily removed from this system. Otherwise, the pH will drop down and everything will come to a halt. And fermentation may also hamper. In long term, the animal may also die. So, for a safer perspective, these gases are being removed through the eructations or the belching through the oral route. That is only the way. They cannot also pass through the lower tract to small intestine and so on. So in that way, if you see, the methanogenesis is a beneficial process for the livestock, because it is removing the fatal gas like hydrogen away from the rumen. So you can say it is a metabolic necessity. Then why I'm saying it is a waste process? And these are some factors which affect the extent of methanogenesis. More the feed animal consume, more would be the methane productions. If the organic matter is more would be the methane productions. But it is not only absolute, it also depends on type of the microbiota prevail in the rumen, which is deciding the extent of methanogenesis. So there are many more factors which are impacting the overall emissions of the enteric methane emission from livestock. Then the question comes, is only the methanogenesis a single way to remove this hydrogen and CO2 in the form of methane? No, there are other ways also. This is the methanogenesis, which is the most prevailing in the rumen. But there are other ways like acetogenesis, sulfate reduction, nitrate reductions, and the propionegenesis or propionate production, through which the CO2 and hydrogen can also be removed. Now the question comes, if there are other pathways like acetogenesis, sulfate, nitrate reductions, then why the nature or these livestock are taking a pathway which is not beneficial or leading to climate change, leading to the energy loss? So here is the answer. You can see why the methanogenesis is a most preferred or favorable pathway. It is the thermodynamics, dear participants, which is deciding the fate of a reaction in the rumen. These are the hydrogenotropic pathway, as I told, because they are removing or utilizing hydrogen. So this is delta G refers to the Gibbs free energy. So more the negative Gibbs free energy will decide whether this reaction should take place or not. If you compare the sulfate reduction and methane reduction, the sulfate reduction, because of more free Gibbs free energy, should be a favorable reaction in the rumen as compared to the methane. But that's not the case. It is not only thermodynamics. It is the end product also, which is through feedback inhibitions deciding the pathway. So here the end product is hydrogen sulfide. This hydrogen sulfide, it is a fatal and extremely hazardous chemical for the body. So that is why, because the end product, this sulfate reduction is not a prevailing or the major pathway for the hydrogen removal in the rumen. So that is why the methanogenesis is taking place or prevailing the for the hydrogen removal in the rumen. If you compare the methane and reductive acetogenesis, here the thermodynamics plays an important role because of the less energy dispensation in the system. This reductive acetogenesis is not a prevailing pathway. Now I'm moving to the mitigation approaches. What are the mitigation approaches we have developed? There are many. Basically, I'll not be talking about all. There are many since 90s. The researchers globally are working on the various approaches, and there are some of these approaches are extremely successful, and some are extremely, the failed under failed conditions or in the long term. So if you categorize them, they basically belong to the dietary or chemical based approaches, or the plant or secondary metabolite based approaches, or the biological approaches. I'll be talking about few of them. Yeah. In the tropical tree leaves, if you see, you know, our main focus should to develop such approach which have basically the field application. Number two, it should not be very high input cost demanding. Otherwise, there'll be no adoption of your mitigation approaches in the field conditions at all. So at NINP, we tried to evaluate some tropical feed resources. Basically, we have one project funded from the ICR, and this project is in place since 2007. It is a network project on methane. So almost 15 years we have completed for that project, and we have evaluated more than 1500 feed ingredients for their methane production potentials, and also conducted some animal studies in large and small ruminants. So here you can see, based on our research, we have finally come out with three tropical tree leaves: banyan, jackfruit, and neem, which are abundantly available in India. So when we used these items at the rate of 10% of the basal diet, there was a reduction of around 20 to 26% in daily enteric methane emissions. So this could be one approach which can be directly taken to the field conditions, and it can be implemented in any of the states, in any of the species. Another experiment what we have conducted was from the Assam, and the Macellus bombicina, the local name is the soap, and the other source was the Cesium cumini, which is nothing else rather jamun. So we have tested these two sources and found a significant reduction in the enteric methane emissions. And basically, if you see the tropical tree leaves like neem, banyan, jackfruit, or the Macellus bombicina, or som, or jamun, what they have? Basically, they have some secondary metabolites. The majorly is the tannin. And tannin also, what kind of tannin? The condensed tannin, which is mostly accountable for the reduction in methane emissions through an inhibition of the rumen protozoa. So first you should understand what these protozoa do, because this tannin is impacting the protozoa, and these protozoa transfer the hydrogen to methanogens, which by using CO2 and hydrogen, combining and converting it into methane. That means the protozoa in the rumen has a role of like a middleman. They are taking hydrogen from bacteria and giving it to the methanogen. So if you cut the middleman, there will be no direct relations between the hydrogen producing microbe and the archaea or the methanogens. The methanogens are incapable. They cannot use the hydrogen directly. They need some middle source for the transfer. So anyway, so here is one product called Haritadhara, which we have developed here at NNP. It is an anti-methanogenic feed supplement, and we have conducted the in vitro and in vivo study, and it shows a reduction of up to 50% if you use at a 10% level. But at this level, at highest level, we also found some reduction in the dry matter digestibility, which is not recommendable, or any reduction in methane emission should not with the concurrent reduction in the feed available nutrient uh, dispensation. So therefore, we are recommending the use of Haridwara at this level, where around 20 to 25% reduction has been achieved. And this is the patent which is being filed for this Haridwara. And this is what I was talking about from the animal studies in small and large ruminant, around 22%, or anything, we cannot directly say that 22. There should be always a range, like 20 to 25% reductions is always possible with the use of Haridwara on daily enteric methane emissions. And this product was launched basically in 2018 by the then Honorable Agriculture Minister, and there were some other ministers, central ministers, and DG, and our director. And basically, this Haridwara technology is commercialized to three farms. One is Saideep Exports Private Limited. This Saideep is producing 10,000 tons of the animal feed every month. And the second, it is also being commercialized to the carbon trading firm or carbon crediting. And apart from that, for field application, the technology is also commercialized to the BAF, which has the units into 7 states. So through BAF, it will also reach to the dairy farmers. Apart from that, we also developed another strategy where the Tamarin Plus, we have one patent obtained for this Tamarin Plus and published the findings in various journals. The use of tamarind seed dust, tamarind seed dust is also a potent source of the condensed tannin. And if you feed tamarind seed husk at a recommended level of around 2.5 to 5%, you can reduce the methane emissions by 20%. Here one question comes, what should be our target? How much we should inhibit the methane emissions in the animals? Should we go for 50% or 80% or 5%? Basically, as you know, the methanogenesis or methane generation is for the metabolic continuation in the environment. If you inhibit it beyond a recommended or desirable limit, what will happen? The hydrogen will start accumulating. So rather than having the beneficial impact, it will adversely affect the animals. So there should be some desirable limit, and which is around 20 to 25 or maximum up to 30%. Beyond that, you should never make an attempt to reduce it further. So another approach which we tried was the silkworm pupae. It is purely sort of unconventional type of the feed material, and we have tried the 2% of the basal diet, and also filed a patent for this source. Basically, people have worked on silkworm pupae meal as a protein source, but we were first to work on the silkworm pupae oil, and then explored its composition in terms of the fat and other factors. And we evaluated the silkworm pupae oil at different levels, and in short-term supplementation, long-term supplementation, the continuous supplementation every day, and intermittent supplementation 1 week on and 1 week break. So we have evaluated this oil source in short, in small ruminants, in large ruminants, and our findings revealed that it is a very potent source for inhibiting the methane emissions by 16 to 18%, and through a continuous supplementation. If you look at here, the withdrawal, we have given the oil up to 6 months. Then we have withdrawn the oil from the diet. And here you can see within 30 days of withdrawal, the animals in the treatment group were at par with the control group. And that means for the effective and long-term uh, uh, reduction in daily methane emissions, you have to keep supplementing the oil. Once you withdraw, within a short period, the animals can come back to the normal level of emissions. Then we also tried to compare the cattle versus buffaloes in terms of the daily enteric methane emissions. There are various questions we are receiving from the parliament on regular basis. Is there any difference between cattle and buffaloes? And some of the studies have been conducted previously in India, but not at the same farm, using same environmental conditions and on same feeding regimes. What we used to do, like somebody has conducted some experiment at IVRI in cattle and the other experiment in buffaloes at NDRI, we used to compare, but that's not a very authentic way of comparing the results. So under one international project, we had one objective to compare the methane emissions between cattle and buffaloes. When the diet is same, environmental conditions are same. So we have conducted the experiment in a series by taking different diets. Here you can see the diet is just reverse, and here also concentrate and roughage. Here roughage, I mean dry roughage and green fodder and concentrate. So all these studies revealed that there is absolutely no difference in the methane emissions per unit of intake. I'm not saying the buffaloes are generally heavier than the cattle and their intake is more. So definitely the methane emissions will also be more. But when we compare per kg of dry matter intake or per unit of intake, it is absolutely similar between these two species. But apart, that means the host doesn't have any control or regulatory mechanism between the host species. The cattle versus buffaloes, they are same in terms of emitting methane emissions per unit of intake. But if you compare the diet, the high roughage or high concentrate, here you can see it is around 27 gram and here it is 24. That means 4 to 5 gram of more methane is being emitted for 1 kg intake. And for example, if you see, suppose an adult cattle or buffaloes, they are eating 10 kg of dry matter intake, and there is a difference of 4 kg between these diet. So there will be around 40 gram of the more methane emission on high roughage diet in one day from one animal. So this is mostly the impact of diet on the methane emissions. And here you can see we also try to compare the ruminal microbiota between cattle and buffaloes. And most of these microbiota you can see were almost similar between these two species. Some of the differences what we found within a very limited fractions, particularly the methane utilizing methanogens like group 3, group 4, group 9, group 12. These are the methylotropic methanogens which use methanol as a substrate, not CO2 as a substrate for producing the methane emissions. Apart from these feeding based approaches, the breeding could also be another option for lower methane emissions. Although it is very in infancy stage, you can see only 1 or 2 studies have been conducted. At the New South Wales in Australia, they have done one study where they produce the progeny from the efficient animals and low efficient animal, and the progeny from the efficient animals. Efficient means the feed conversion efficiency I am talking about. The animal which has high feed conversion efficiency will definitely produce less methane as compared to the animal which has very low feed conversion efficiency. So around 24% of the less methane was produced by the animals which comes from a superior bull as compared to the inferior bull. But still there is lot need to be done to to establish that the methane emission is also a heritable trait. It is not yet established. The people in New Zealand, like Ag Research Institute, they have studied, but the heritability estimate was not very high. So it is yet to be established. So now come to the benefits and implications after doing all these inhibitions or reduction in methane emissions, what the farmers is going to get or what would be the benefits or implications at the country level. We can see here. As I told, there is around 38 into 10 to the power 8 giga energy loss from the dairy animals. And if you convert that loss in terms of the money, around 48,000 crores is being lost from the farmers just due to the methane emissions from dairy animals. So we need to focus to minimize. I'm not saying you cannot bring down it to zero because you cannot inhibit the methane emissions 100%. But simply by targeting 15, 20 or 25%, you can definitely bring down this loss. So if you are targeting around 20%, what would be the impact? The total digestible nutrient, there is a deficiency of TDN. If you see the various estimates given by various agencies in India, there is a deficit of 23% in total digestible nutrient availability. So if you simply reduce the methane emissions, this deficiency could be bridged by around 7 to 8% simply by reducing the methane emissions. So if you see by reducing methane emissions, what basically you are doing, you are saving some biological energy. And this biological energy can be used somewhere in the productive functions by the host animals. So the milk productions can be increased on an average by 16 million metric ton from the existing one just by reducing the methane emissions. And these are some monetary benefits. How much it would be? The current total milk worth is around 7,000 billion, which could be as high as 7,700 billion by reducing this greenhouse gas. The current milk availability is around 360 or 65 gram, which can also be increased by 30 gram, and it could go up to 395 gram just by minimizing the GHG emissions from the dairy animals. So in conclusion, what we can conclude that our major focus should be on the production of climate resilient, because you cannot stop all of a sudden the change in the climate change. As I told, it is a long-term phenomena. So we need to majorly focus on those crop and livestock species which are climate resilient and have the adaptation mechanism to cope up with the stress conditions without compromising the productivity. And methane emissions, as I told, is an adaptive mechanism. It removes hydrogen away from the rumen. So you cannot stop this mechanism also. Rather, you can reduce or curtail this pathway by 20 to 25% maximum. And this reduction should be practically feasible, where the fermentation and nutrient availability to the livestock remain unaffected. And we primarily need to focus on those approaches which are farmers friendly. Please do not recommend them anything which is not feasible to them. It should not be very costly, and it should be minimum input cost demanding, and the resources or the raw material should be available within the nearby vicinity. Then only the farmers friendly approaches could make some implications in the field conditions, otherwise not. And we need to evaluate any product or strategies or technologies in different species in field conditions before recommending it for the taken up.